Hello everyone, welcome to the Dean's Lecture Series. It is a real pleasure today to have Devika Chithrani here from the Department of Physics and Astronomy and talking to you about gold nanoparticles as magic bullets to treat cancer. My name is Peter Luk, I'm the Dean of the Faculty of Science and the Department of Physics and Astronomy is one of 6 departments in the faculty. It is a real pleasure to have Dr. Chithrani here. A few words about her biography. She received her undergraduate degree from the University of Colombo in Sri Lanka, then moved to Toronto for graduate degrees, Master of Science at U of T, University of Toronto, PhD degree in 2004, also from the University of Toronto in physics. And then she spent some time as a scientific associate at the Princess Margaret Cancer Center in Toronto, then as a staff scientist at St. Michael's Hospital in Toronto, and then finally as an assistant professor at Ryerson University in Toronto, now called the Toronto Metropolitan University, of course. Since 2016, she is at the University of Victoria in the physics department, quickly rose through the ranks. She's now an associate professor here and is working in medical physics, so broadly in the application of nanoparticles and nanoscience to the treatment of diseases, in particular cancer. Dr. Chithrani has been very successful in her research. Her papers received 11,000 citations. Her work can be found on covers of journals. She has been awarded the Faculty Gold Medal and the Gold Medal for Physics when she received her bachelor's degree, and has been the recipient of several fellowships by NSERC, so the Natural Sciences and Engineering Research Council of Canada, for her graduate and postdoctoral work. She's been ranked among the top 2% of scientists in the world by a database maintained by Stanford University. And again, her passion is it to develop smart nanomaterials to treat or improve cancer therapeutics. Just a few words about the field of cancer treatment. Of course, that is an old field, but it becomes more and more important. By 2040, so in 17 short years, the number of new cancer patients per year is expected to rise to 29.5 million annually, and the number of cancer-related deaths to 16.4 million. Approximately 50% of all cancer patients can benefit from radiotherapy, but radiotherapy is not very specific. It doesn't actually treat the tumor directly. The current methods need improvement to increase patient survival and to treat tumors more effectively. And so this is where Dr. Chithrani comes in. She uses gold nanoparticles as a radiation sensitizing agent to optimize the current radiotherapies. And in the talk we're going to hear, she will discuss the importance of using gold nanoparticle-based strategies to overcome all these challenges related to tumor treatment in their particular microenvironment. So without further ado, over to Dr. Chithrani. Thank you very much.
First of all, I would like to thank our Dean Peter for that great introduction. Today, I would like to share my 1.5 decades of research with you all. And my research is more focused towards improving current radiation therapy. If you look at current treatment modalities for cancer treatment, radiation therapy is one of the major ones. So the question is, what are the problems of current cancer radiotherapy? So in my program, I am trying to address one of the problems that we are having in current radiotherapy. That is to use gold nanoparticles as a magic bullet. So when we talk about gold, you always think gold is yellow. But when we make nanoparticles, it actually, you can see it changes color. So nanoparticles are different compared to bulk gold that we know. So the most interesting thing here is that we can actually use these gold nanoparticles in a very innovative way to treat, to actually improve current cancer radiotherapy. So the endless possibilities of this nanotechnology was introduced by our former Nobel laureate Dr. Richard Feynman. He said that actually in a talk at Caltech in 1959, saying that there is a plenty of room at the bottom. Actually, this officially started the advance of nanotechnology. So, to give you a small introduction as to what is the scale, what is nano? It is very, very small if you want to think about it, and if you take a drop of blood, it has actually 5 million red blood cells. If you look at the diameter of a red blood cell, is 2500 nanometer, and if you look at the width of a DNA double strand, is 2 nanometer. So, you have an idea how small this nanoscale, and you wonder how it can make such a big difference in cancer therapy. So, that is what I am going to talk about. What is the role of nanotechnology in cancer medicine? Before I do that, I want to show you a diagram or schematic of a cell. We are made out of trillions of these cells. So, these cells have special organelles, nucleus, membrane, and few other organelles, so to function, right. And so, now if you look at what is the dimension of a cell is 10 micrometers across, and if you look at what we eat, proteins most of the time, right, proteins are around 10 nanometer scale, and if you think about, you get sick with viruses, especially COVID-19. So, what is the size of a COVID-19 virus? It is about 100 nanometer. So, if you look at proteins that we take is about 10 nanometer, and the viruses are about 100 nanometer, they both can get into cells. So, if we make nanoparticles in this range, we can actually send these nanoparticles especially to cancer cells and to treat them in a way, so that we can actually battle this cancer in a more innovative way. So, what are the platforms that we have right now? There are many nanotechnology platforms available. Out of all of these, I would be focusing on metal nanoparticle platform, because I will be focusing on gold. Gold is a metal nanoparticles. So, now you wonder how these nanoparticle transport through our cells. So, it is very interesting. So, then these nanoparticles can, so the marked in red are gold nanoparticles. So, they actually can get into cells, and then they can get trapped in these small organelles in the cells, and they get processed and they get excreted at the end. So, this is a schematic. How do they look like in a real cell? So, if you look at it, in the middle looks like a hole, is the nucleus of the cell. That is the biggest one in the cell, is nucleus. It is basically brain of the cell. And so, you can see small red clusters there, those are gold nanoparticles, right. And what you see in green basically are microtubules in the cells. These are basically highways in the cells. So, these nanoparticles can traffic through these highways. So, now the question we have is of course, we can use nanoparticle. One of the questions that I am trying to address is will the size of these nanoparticle really matters? So, in order to do that, what you need to do is you have to make different size nanoparticles. So, you can go from 10 to 100 nanometer, that is our goal, right. So, the question is, what material system allow you to do this kind of research? One of the materials easily you can do is with gold. In that, what you do is you change the gold salt to the reducing agent, the ratio, and you can end up with different size gold. So, you actually can analyze what is the effect of size of gold nanoparticles, how they get into the cells. So, what do you think? You think will it make a big difference 10 to 100 nanometer? It is not that you may think it may not make a difference, but let us find out. So, what I did was so these nanoparticle transport across the cells, will the size matters? Yeah, that is what you can see to the right hand side, you can see a graph, you can see there is a dependence, right. So, small and large, like small means 10 nanometer, large mean 100 are taken up lower compared to 50 nanometer gold. That is interesting. That means this, even though it is a smaller scale, size can play a big role, that you will see later in my talk as well. So, now the question is, this is gold nanoparticles has a size dependence, but what I am interested more is more applied science. So, when you think about gold, what do you think about gold? It's a high atomic number material, right. So, what do you mean by high atomic number material? That means it has lots of electrons. So, the idea here is when patients are treated with radiation, what you have is the photons, right. We use photons for radiation therapy. These photons actually can kick some of those electrons out and create a shower of secondary electrons. So, what happens with this shower of secondary electrons? So, what they can do is they can actually interact the water molecules in the cells. So, if you look at it, 90% of our body is actually water. So, the water molecules interact with these secondary electrons can produce something called free radicals. These are damaging species. That is what we are told, like eat apples, because that actually can reduce the number of free radicals. That is why we are asked to reduce the number of free radicals, right. So, these free radicals, enhancement of free radicals by gold nanoparticles in a tumor cell can actually kill the tumor cells. So, that is the theory behind using gold nanoparticles for cancer therapy. Now, when you want to talk about cancer therapy, think about it this way, the lesion in the middle marked more like a reddish color is the tumor. So, the question here is how do we treat this tumor? So, in order to do that, you have to send photons to this tumor to kill the tumor. That is what you do in a radiation therapy. So, the question is, how do you reduce the toxicity to surrounding tissue, right, around the tumor? So, in order to do that, what we do is if you can see that LINAC image to the right, you can see that can rotate. While rotating that, you can actually rotate the radiation beam in a way it crosses the normal tissue one time, but it crosses the tumor every time. So, that is the theory behind. Now, if you look at it further, what you can see a graph here. The dotted line to the most right one is basically normal tissue toxicity graph, right, and the one middle is how we can control the tumor. Now, I am going to show you something here. So, if you think about it, if you want to have the 80% tumor control, what you need to do, so you can still see, look, please focus on the two graphs to the right. So, what you see is when you have to control 80% of the tumor, you still see significant amount of normal tissue toxicity. So, the question is, how do you reduce that? In order to do that, what we do, we actually put gold in the tumor. So, when you put gold in the tumor, what we can do is we can really locally enhance the radiation dose. So, you can see what we can do, we can shift that curve in the middle to the left. Now, if you look at normal tissue toxicity curves, which is at the far right, you can see at the 80% tumor control, normal tissue toxicity is much less. So, this is the idea behind gold nanoparticles to treat cancer therapy. Now, let us see, is it being done, if it is that good. So, yes, it has been tested. So, if you look at this curve, this graph, you can see the curve at the bottom, that is, if you treat the tumor with gold nanoparticles and radiation, you can actually reduce the tumor volume significantly. But this is done in 2004. So, now this is so it has been more than how many years now, right? But why it is not translated into the clinic? That is a problem, right. So, that is what my research is trying to focus on, how you can get better. So, if you think about this study, they use 1.9 nanometer gold. Now, I want you to go back to my fundamental study, where I showed you very small gold nanoparticles have a very low uptake. So, in this case, they have to use a very high concentration of gold, which is not clinically feasible. But if you want to bring something to the clinic, you have to reduce that concentration significantly. So, what I did was I optimized the size of the gold nanoparticles and then I was able to reduce the concentration that you need by a factor of 1000, and then it is clinically feasible. That is what my work is bringing to the table right now. And so, I will show you some of the results now. So, I use 14 nanometer, 50 nanometer and 74 nanometer, and this is actually showing, you can see gold nanoparticles are incubated in the cells. You can see orthovoltage unit, that is what the treatment unit for superficial radiation in terms of cancer therapy. And what we noticed was something very interesting. You can see this is the survival of cells, right, on the y axis, x axis is the radiation dose given. So, if you look at the 10 gray dose all the way to the right, you can see the red curve where you can see actually that is the cells treated with 50 nanometer gold has the lowest survival. So, which is what we needed, right, because they are going into the cells more. If you remember 50 nanometer particles go in more to the cells. By doing so, it can actually damage cells more. So, this is survival of cells. So, the question is, we always talk about DNA damage. So, what happened to the DNA damage, which we talk about in the presence of the gold? You can create more free radicals that damage DNA. Can you see that in your research as well? That is what you will see now. You can see the top panel is actually the cells with no gold nanoparticles, the bottom panel is cells with gold. So, these you can see small dots, those are actually damaged DNA. So, you can see there is more damaged DNA in the cells treated with gold. So, with this what we see. But then I have another question. So, this treatment unit is used basically for superficial, that means closer to the surface tumors. You cannot penetrate deep into the tumor. So, the question is, will your technology work in a setting like that? That is what I have. This is a linear accelerator which you can actually use to treat patients where the tumor is deep seated. So, the question is, will my technology work even at that mega electron volt energies? Yes, it did. So, you can actually see the top row like before is the cells treated with no gold, the bottom one is cells treated with gold. So, you can see this number of this damaged DNA, though the foci these dots are foci that represent DNA damage. So, you can see more DNA damage with gold nanoparticles even with mega electron volt energies that is used in the clinic. So, when we did this research, it was so significant that it was featured in the Radiation Research Journal, which was the premier journal of Radiation Research Society. And it was awarded the Michael Patterson Publication Award for the significance of that finding, and because it makes a big difference in the field. Now, I want to summarize what we have done so far. So, that means we actually showed you size of the nanoparticle really matters, and we also show that gold nanoparticles can actually can use as a radiosensitizing agent. You can see it enhances the DNA damage. This is what we want. But all of this work is done on in 2D, that is a monolayer of cells. But in real life, this is not the case, right? In patients, you have a 3D tumor, it's a 3-dimensional tumor, is a 3-dimensional structure. So, the question is, how do you translate this kind of work to a model where it is much closer to the patient tumors? So, in that case, what we did was we started developing 3-dimensional tissue models which actually can mimic real tumors in patients. So, why I am so interested in these things is that if you think about it, so say if I want to treat these patients with gold nanoparticles, you need to inject these gold nanoparticles intravenously. So, what happens when you inject them? It goes through the blood vessels, and then it goes through the blood vessels and it enters the tumor blood vessels. So, what is the difference between tumor blood vessels and our normal blood vessels? The tumor blood vessels has a unique feature where, because they are formed rapidly, they have some weaknesses. That means there are some places where these nanoparticles can leak into the tumor tissue. That is what we are capitalizing on. in tumors to use this nanotechnology. So, the question here is how these nanoparticles behave in a 3D tumor model before we testing in an animal models, because my rationale is I do not want to use these technologies in an animal models unless I am optimize these things in a 3D models, which is much closer to tumor models, because that allows you to do more interesting science, at the same time use less number of animals. So, we actually built two different types of tumor models. In one case we did was we grow layer by layer of cells, and then you can see in the middle image you can see a cross section of those tumor cells. You can see layer by layer tumor cells. So, this is actually a 3D tumor. And if you, so we use another method where we make tumor spheroid. You can see in the middle image of the right hand, it is a tumor spheroid, it is like a 3D tumor. And so, we created these two models, and all these models are published, and so we use these two models to find out whether this nanoparticle size really matters in a 3D tumor environment as well as we did see in a 2D environment, 2-dimensional. So, I just wanted to show you graphically what is the outcome first. So, if you look at the left hand corner, you can see is the monolayer study, that means 2-dimensional. You can see smaller nanoparticles are taken up less compared to larger nanoparticles. But on the right hand side, what you see here there is a 3D tumor. So, you really can see the difference. What is the difference you see? In a 3D tumor you have more barriers. Do you see that? There are more barriers. So, that means small nanoparticles as a result could penetrate better than larger nanoparticles. So, now I will show you real results. So, in a monolayer of cells you can see small nanoparticles are taken up less compared to larger like 50 nanometer, but when I go to tumor tissue you really can see the barrier in between cells, right. In the monolayer you do not see that. So, that affects how these nanoparticles go through. So, when you look at the experiment, you can see the graph now actually smaller to the graph to the right, and you can see for two cell lines you can see small nanoparticles are actually better than larger nanoparticles. But then it is not the same thing with the monolayer, right. So, the question is how do you get around this thing. So, in order to do that, what we did was we did some changes to the surface of the gold for smaller nanoparticles, because now we know because gold nanoparticles penetrate better in actually 3D tumors and as well as in vivo. So, what you can see here is we actually changed the surface in a way. So, it actually has a very high uptake in 2D monolayer to the left hand side, to the right hand side as in 3D tumors as well. So, which is good. Now, what you do next? Okay, so it works for 2-dimensional, that means at single cell level, and then it works in 3-dimensional level. Now, you want to see it in an animal model. That is what we did, because we optimize it already. So, then you inject this gold into an animal model, like you actually create a tumor at the back of the mouse, you can see subcutaneously, and then you inject these gold nanoparticles to the mouse, and then these gold nanoparticles can go through the blood vessels, enter tumor blood vessels and enter the tumor tissue. So, we actually look at as a function of time how these nanoparticles accumulate in the tumor. So, you can see within 24 hours you really can see a higher uptake of gold nanoparticle in the tumor. So, if you want to see where these gold, this is a tumor cross section, you can see a blood vessel with gold in the blood vessel, you really can see it lit up with gold, right. So, you really can actually target these gold nanoparticles into the tumor. So, now, I want to see now what happens after that. So, now we know we can accumulate gold in the tumor. If you look at it now, we give a radiation dose of 5 gray to the tumor, and you can really see now after 28 days you really can see there is a slower growth when the tumor is treated with gold nanoparticles and radiation therapy. But you may think this may not be that impressive, but then how can you make it even better? So, that is what I am actually looking into now. So, in order to do that, you have to be smarter. So, what I meant here is that I am going to use some of the chemotherapy drugs in combination with radiation therapy and gold nanoparticle. In other words, you can say you can repurpose some of the clinically approved drug to improve current radiation therapy. So, one of the drugs that I used was docetaxel, which is a very useful drug in the clinic for patients with metastatic tumors. It is used heavily. So, why do I want to use gold? Why do I want to use docetaxel? There are a couple of reasons to do that. One is, it actually, if you look at that cell cycle, the dotted line shows how the cell cycle population varies in a normal cell, right? But when you treated these cells with docetaxel, you can see it actually changes the cell cycle, the darker curve. So, the solid line curve, you can see it actually put most of the cells in a phase called G2M. Actually, that is the phase which is most sensitive to radiation. Now, you think about it. So, the drug can put cells in a phase which is most sensitive to radiation, and you want to give the radiation dose to these cells. So, it is vulnerable and we can capitalize on it. So, what is the beauty here with the gold nanoparticle and docetaxel is amazing. Now, for example, if you look at the left hand image, you can see the gold is distributed throughout the cells. Now, imagine if you can bring all of this gold closer to the nucleus of the cells. Let us see, that is exactly what you can do with this drug. Now, if you have radiation, what it does? It actually can produce a shower of secondary lectin to the nucleus and can damage the nucleus. Isn't that fantastic? So, that's what we see. So, the top left hand, it shows with the treatment of the docetaxel, you actually can have more gold nanoparticle in the cells, which is a plus thing because gold is a radiosensitizer. It is good to have more. And it's another very important thing is the bottom graph shows it actually allows us to retain the gold for a long time. So, which is also important because the radiation therapy is given as a fraction. That means the patients are coming to the clinic every day for 5 days to get 2 gray of fraction if it is a fractionated dose, right. So, this is having gold inside the cells for a long time can help patients not to have this dose every day. So, now this actually shows we use the radiation, we actually did the radiation treatment. What you see is to the right hand side, it gives a synergistic therapeutic effects, gold nanoparticles with docetaxel, fantastic. So, this is actually it's still on 2-dimensional level, right. And then what we wanted to do is as you said, we built 3-dimensional levels, the models. And what we saw was it's amazing, it's similar to what we see in 2-dimensional, that is a single cell level. Even in 3D level, that means the tissue like level. So, you really can see there is a more than 100% increase in gold nanoparticle in 3D tumors as well, 3D tissue models, which is fantastic. And in addition, what we saw was in terms of radiation therapy, you can see it's actually with the treatment of the docetaxel and gold nanoparticle and radiation, and you can see how good the therapeutic effects are. Now, the next question is how this will affect actually in vivo. That is the $1 billion question, right. Will you provide similar results in an animal models? That is what we are going to find out. So, this is we tested gold nanoparticle and docetaxel in an animal model before we do radiation first to see whether the gold can actually get into the tumor. Fantastic, you see over 100% accumulation of gold in the tumor even in vivo. So, which is fantastic, and do you believe it? So, then you can see the graph. So, you can see the gold is marked in yellow there. So, the left hand one is treated with just the gold, the right hand side you can see treated with gold nanoparticle and docetaxel. You definitely can see more gold accumulated in the tumor treated with docetaxel and gold, and it's amazing. So, now what is next? Next is actually to test the radiation experiment, right. You have to treat the mouse tumor with radiation. So, So one of the things we talk about is the effect of this drug has on the cell cycle. So we want to know, before we do radiation, we actually check whether even animal tumors are actually in a way that it is actually more geared towards G2M phase, which we talked to you about. That is, it is the most sensitive phase to radiation. What did we saw was amazing, that we saw with the treatment of docetaxel, even in animal tumors, you can see the most of the tumor cell population is in G2M, which is G2M phase, which is fantastic. And then what we did was we actually delivered the radiation dose. This is what you see actually. You can see actually here big improvement of the tumor control with the treatment of gold nanoparticle radiation and docetaxel. So this is our highlight of my work right now, that is the combination of gold nanoparticles and docetaxel is beneficial. Now we are moving on to other bigger studies, so that we can actually get into a clinical trial sooner. Now I am going to change the gears a little bit. So for example, when you talk about cancer, are we talking about only cancer cells? No. We have to think about the tumor microenvironment. So what about the tumor microenvironment? So in every tumor that we have, there is something called fibroblast cells. They are not cancer cells, but what the tumor cells do is actually transform these normal fibroblasts into something called cancer associated fibroblasts. We call it CAF. So there is a big difference between these normal fibroblasts and CAFs. So these CAF actually promote tumor growth. So now I want you to think about it. Now if CAF promote tumor growth, you really want to know how these gold nanoparticles would interact with these CAFs as well, because otherwise if it is not effective, then it will, if one tumor cell survive, this CAF will promote that tumor growth, and then it will happen again, recurrence of tumor, right. We want to avoid that too. That is what I wanted to find out. And what you can see here is it was very interesting. What we saw was if you look at the CAF, is the cancer associated fibroblasts take up so much gold. So if you look at to the right hand side in the image, this is CAF, they are much bigger. You can see how much gold that they take. So the left hand image is the left hand corner image is actually the tumor cells which take up gold too, but the middle one is normal fibroblast. So they are not taking up more gold. So now the question is, can we use these gold nanoparticles in these cancer associated fibroblast cells to reduce its activity, so it cannot promote the tumor growth anymore? That is exactly what we want. So can we do that? So we did that. We actually gave radiation. So what we found was we was able to increase the DNA damage in the cancer associated fibroblasts. Actually it cannot promote tumor growth as it used to be, which is a very positive result. But now you have to remember, what we did here was we used cell lines separately, but in a real tumor they coexist. So what we did was we actually did co-culture of these cancer associated fibroblasts and tumor cells, which is a complex experiment to do, but we were able to do it. And then you can see in that image, the CAF is the bigger cell, tumor cells are the smaller cells, right. And then you can see CAF marked in red is the gold. You still can see more gold in the CAF and also in the tumor cells. So after you do this kind of co-culture, you need to separate them, and in order to do, there is a mechanism, we use magnetic beads, but we were able to separate them, and finally we were able to see the amount of gold in the CAF versus the tumor cells. You really can see, even in co-culture you can see more gold in the CAFs. So which actually led us to do a very neat experiment where we did radiation co-culture. We could really see we were able to reduce the resistance of CAF to therapeutic effect, so that we still could see significant benefits of radiation with gold nanoparticles in a treatment process. So this is very important, and so this is what we are doing right now in animal. We are creating animal models in a way that it actually mimics this kind of scenario where we have this cancer associated fibroblasts and tumor cells, all of these things. The reason I am telling you is because if you use standard cell lines, you usually don't see this kind of tumor environment, right? And that is why some of the therapeutics fail at the end of the day, because they just studied the solid tumor models where you do not see these CAFs and other cells. So, they actually may play a huge role in this kind of therapeutic approaches. So, now final lag is about what else can you do. So, now I talked to you about gold nanoparticles, docetaxel, how you can use them together to improve current cancer therapy. So, but in those experiments, we injected gold and the drug separately, simultaneously, but then the question here is can you deliver them simultaneously to the tumor? In order to do that, we actually created a vehicle where it is a lipid nanoparticle system, and where you can put gold nanoparticles and the drug inside this system. So, it can be done. So, this is just to show you those circles are lipid nanoparticles. You can really see actually gold nanoparticles inside the cells. We are actually collaborating with the industry partner to do this kind of research, because its technology is high end. So, we need support from industry. At the same time, for translational purposes, it is important to have partners. And here I show you how the use this kind of system actually allows us to actually send more goals to the tumor environment. So, which is a plus thing. Plus, in this case, then you do not have to send the drug everywhere, because it is encapsulated. So, that allows us to reduce the side effects to the patient. So, these are a couple of platforms that we are developing in this lab, and so things are moving forward, and it's a lot of progress. And how can I do it by myself? No, I have a lot of collaborations with a lot of international collaborators. So, we always talk, we have meetings, and we actually wrote a roadmap how we can translate this kind of technology to the clinic. So, clinicians are involved, and so that is how we do things. We collaborate heavily with other partners around the world, and these are my heroes and my undergraduate students, and a lot of funding from different granting agencies. I am very grateful, and also a lot of collaborators. The bottom row is all clinicians, radiation oncologists, medical oncologists, pathologists. At the bottom row, the top row actually, the first one is Nancy Dos Santos, doctor, so who is actually does a lot of animal work with us, and our industry partner is Chris Tam, and all the other people we have is the medical physics, biochemistry, and drug delivery experts in that team. And at the end, I just want to show you my final contribution other than research is my daughter, who is going to be a clinician scientist. This is actually a picture I took when I went to Disney World. She was very little. She wanted to be a clinician scientist, and so I am happy to see that that dream is coming through. She is in the second year of medical school, so very proud of her. And so, I moved from Toronto to Victoria a few years ago, and happy to live in Victoria and in this beautiful campus, and I do not think I can find a campus like this anywhere in this world. So, I am so happy to be here. So, thank you so much for listening.